Good morning, dear students. I am Rakesh Patel from RG Shah Science College, Ahmedabad, and with me on my extreme left is Mr. Mukul Upadhyay, Mr. Kunal Upadhyay from Gnanganga College of Science and Management, Rajkot, and besides me is Dr. Vasant Jadeja. She is from MNN Virani Science College, Rajkot. Both have vast experience in the field of microbiology and have been teaching the microbiology for more than a decade. And today they are with me to discuss about the Operon model. Now, what is an Operon model? If we want to study that, if we want to discuss that in detail, before that, let me take you to a basics of genetics in short. All of you know that the basis of hereditary in a living cell is a genome. That is, in eukaryotes we call it as a chromosome, while in case of prokaryotes we call it simply as a genome. Now, this genome contains the complete set of hereditary information. These genomes have various genes on different loci at different sites, and these genes, you can say that they are the sequences of DNA which specifies the sequence of an independent product. That is, a gene specifies for a sequence of an independent product. That product can be either protein in most of the cases, while in few cases it can be a RNA also. Now, the genome of various microorganisms and other eukaryotes that varies in size. It ranges from 0.6 into 10⁶ base pairs to, for example, in human beings it is as large as 3.3 into 10⁹ base pairs long. So the vastness of genome is too complicated. It is very vast. In case of small organisms, the size of the genome is small and may specify, for example, in E. coli, it may code for or it may encounter around 4,200 genes, while in case of human beings, the number of genes are as high as 23,000. Now, all these genes, they do not function together. All these genes, they do not start coding for a protein or a RNA altogether. They are switched off and switched on as per the requirements of the cell. Cell ne jare jare je vastu ni jarur pade છે એ વખતે એ વસ્તુ synthesize કરશે. Otherwise, it will stop it. This is basically to save the various macromolecules and as well as to save the various kinds of procedures which involves a lot of ATP, a requirement of energy. And if this coordination is not maintained, the cell will not be able to cope up with the systems functioning in the cells. Now, there are certain genes which functions always. We call them as a housekeeping genes, and these genes are always needed. They are always functioning. They always code for a particular protein or a RNA. While other genes, they can be switched off and switched on as per the requirements of the cells. These can be certain kind of genes, like we call them as a luxury genes, or certain genes like, say, inducible genes or repressible genes. Now, the basis of this gene expression, that is the regulation of genes, how these genes are expressed, how these genes are controlled, it can be at various levels, which will be explained to you in detail, but in short, it can be at a level of transcription, or it can be during the processing after the transcription is over, processing of RNA, and even it can be at a level of a translation. But basically, the most fundamental concept is that forwarded by Jacob and Monod in 1961. According to that, there are certain trans-acting proteins which functions together with the cis-acting sequences of the DNA. That is, certain proteins formed by regulatory as well as structural genes, they interact with certain sites on the DNA like promoter, operator, and thereby they carry out the regulation of these genes. How these genes are synthesized? Certain genes are to be induced while certain genes are to be repressed. Under what conditions? All these things in detail will be explained to you by our two professors who have come from the Rajkot. This is a very important aspect as far as the conservation of macromolecules as well as the energy level in the cell is required. The further details will be explained to you by Dr. Vasant. Please, doctor.
Hello. Good morning to all of you, dear students. Lac Operon is very interesting subject, even in the microbiology. Sir has explained that how the induction and repression of the genes takes place. But let us talk in detail that what happens. We know that cell is a very small entity and it carries out number of activities. One of the important activity of the cell is that how to take the nutrition from the environment. The nutrition having a very wide range. We can take an example to explain the Lac Operon, that E. coli, a very tiny cell which consists of a small structure, but it carries out very complicated things. Now in this E. coli, when the nutrition, that is, let us take the example of lactose. So when a E. coli cell comes in contact with the lactose, what happens? A question arises, how this cell is going to utilize this sugar, which type of the enzymes going to degrade this sugar, how the sugar will enter the cell. So all this we can study detail by taking the example of Lac Operon. The most direct way, right, to regulate is the induction and repression of the gene activity, which sir has told us that how the genes are expressed and how the genes are shut off. That is, the switch on and switch off of the gene expression is continuously there, and cell is controlling with the help of the regulatory mechanism. For example, we know that regulation can be done at two levels. One is we can control the activity of enzyme, and second is we can control the synthesis of enzyme. Now let us take the example that enzyme can be regulated by regulating the actions of enzyme. We can take the very basic example of energy link control. You can take the example of feedback inhibition, precursor activation, covalent modification, etc. On the second side, we can say that regulation of enzyme synthesis can be there to control the complete system. That is, the induction of the enzyme synthesis and the repression of the enzyme synthesis. That means the gene expression is controlled by regulation of proteins. In other words, you can say activators and repressor. Both are the protein. The activator is to activate, to induce the synthesis of enzymes which are required for the catabolism or for the utilization of lactose. And whenever cell want to utilize the lactose, it will start the induction of that particular enzyme. So in the case of Lac Operon, we will see that which type of the enzymes are synthesized by which particular type of the genes, right? So in the next slide, we can see the basic, you can say very simple definition of operon, that what is operon. An operon is a functional unit of genomic material containing cluster of genes under the control of a single regulatory signal or promoter. In other words, you can say it's a completely cluster of gene. They are unit or in coordination, they are expressed whenever the cell needs. In the special case, you can say that Lac Operon having a group of genes, but these all are transcribed at the same time. The second important thing is that this usually controls very important biochemical processes, and they are only found in the case of prokaryotes. We know that prokaryote, the best example of the prokaryote is bacterium, and here we have taken the example of E. coli. Now what happens? That there are so many types of operons which are used for different activities. Lac Operon is specific for the catabolism of lactose. That means when the cell is exposed to the environment in which the lactose is present, how it is going to utilize this lactose. That is explained very well by this Operon model, which was proposed by Jacob and Monod in 1961, and they have given the complete model that how it acts and which type of enzymes are produced. Second example, you can say Tryptophan Operon, Arabinose Operon, hot Operon, immunity Operon. But dear student, let us discuss in detail the basic fundamentals of Lac Operon. Now we can see that what happens when this Lac Operon is switched on. We know that genes are there, and these genes, they are expressed according to the need of a cell. Now if the cell want to utilize lactose, so which gene is going to be expressed? That we can study in the case of this Lac Operon. Now this Lac Operon having three important genes which are involved in the processing of the sugar lactose. One of the gene which is responsible to produce a specific enzyme known as beta-galactosidase. This enzyme is generally used for the hydrolysis of lactose into glucose and galactose. The second important gene, even we can see in the common diagram. In this diagram you can see that cap side having a green color code, operator side having the orange color, and between these two there is a promoter side. The three structural genes which are involved in the catabolism of the lactose, that is Lac Z, Lac Y and Lac A. All these three structural genes, they are having the unique functions in the Lac Operon. Now in this diagram we can see, this is the functional diagram which easily explains us that what happens. We know that there is a Lac I. This is a regulatory gene. Then there is a P Lac, that is a promoter region. O is the operator region, and then there are three genes which is known as structural genes, Lac Z, Y and A. Now this, when the cell want to utilize the lactose, this operon gets switched on. That means it will start the process. First of all, what happens, that these three genes which are responsible for the three different enzymes, galactosidase, permease and transacetylase, how it is synthesized. What happens in the next diagram, we can see that all these three enzymes are important because we can see that lactose is present outside the cell, that is in the environment. So how the lactose is going to come inside? That is with the help of specific enzyme known as permease, which is synthesized by the structural gene. What happens, that this lactose will enter inside a bacterium cell, and you can see in the figure that this lactose, with the help of another enzyme which is known as beta-galactosidase, it is converted into allolactose. This allolactose, actually allolactose is the inducer. It is used as a inducer in the case of Lac Operon. It will convert this allolactose to the monosaccharide, that is galactose and glucose, with the help of beta-galactosidase enzyme. But the function of the permease is that it will bring the lactose into the cell. After bringing inside, the beta-galactosidase will proceed, that is the conversion of disaccharide lactose to monosaccharide, and especially the important part is that allolactose, it is formed from the lactose which will induce the Lac Operon. Now what happens, that there are four situations in which this works out. That is, when the glucose is present and lactose is absent, what will happen? The E. coli will not synthesize the beta-galactosidase enzyme, because the organism, that is the cell, will prefer to utilize the monosaccharide glucose because it does not have to break down. It's already monosaccharide. If it take the lactose, it has to degrade into the monosaccharide form. So always the cell will prefer to utilize glucose first, then lactose, if both are present in the medium at the same time. That is, the very first situation is that when there is a glucose with lactose, this enzyme is never synthesized by the Lac Operon. That time the Lac Operon remains switched off because it is never flooded by any extra enzymes, and the cell conserves the energy that way. The second situation is that when the glucose is present and lactose is also present, still the E. coli will not produce the beta-galactosidase. In the third situation, what happens? When glucose is absent and lactose is absent, the cell does not produce it. But in the fourth situation, we can see in the environmental condition, when glucose is absent and lactose is present, the E. coli is going to produce, it will going to synthesize beta-galactosidase enzyme. Why? The reason is that there is no alternate for the cell. No monosaccharide is available, so it has to use the lactose, and so it has to switch on the Lac Operon. Now actually, if we will see that how this proceed in the case of lactose, we can see very easily in this diagram that there is a regulatory gene known as gene I. This gene will synthesize a special protein which is known as repressor. The function of the repressor is to switch off the Lac Operon. Here what happens that this repressor protein, it will come and it will bind on the operator site, and so the RNA polymerase, which is responsible to carry out the synthesis of three important enzymes by the structural genes, cannot sit at its proper site, and so the transcription cannot take place. In the second diagram, we can see that allolactose, that is when the lactose is converted to allolactose, and we know very well that this allolactose, it is an inducer. What happens? This repressor will do the same function, that is it is produced by the repressor gene and it will again binds to the operator site, but now this allolactose, it will bind with the repressor and it will not allow to bind to the operator site. The reason is that the repressor, when binds with this allolactose, it will do a conformational change in the structure of repressor. Because of the change in the structure, now it cannot occupy the place, right? And so that place is available for the RNA polymerase and it can reach to its promoter site and the transcription can proceed. So you can, in other words, you can say the Lac Operon gets switched on. When both the glucose and lactose both are present, then what happens? Now it's very natural that this explains that how the Lac Operon transcribes only when lactose is present. But this does not explain that why operon is not transcribed when both glucose and lactose are present. Now here we can see that when glucose and lactose are present, the RNA polymerase can sit on the promoter site, but it remains unstable and so it keeps on falling. So the process cannot proceed and it gets stopped. In the next diagram, we can see that when glucose is absent and lactose is present, what happens? Now here we can see that one specific protein which is again produced, known as activator protein, because the role is to activate and to stabilize the RNA polymerase, which is very important enzyme to control the complete process. This activator protein works when glucose is absent. In the next diagram, even we can see that what happens in detail. Now this part, this session will be explained in detail by my colleague Mr. Krunal Upadhyay. So I hand over this situation of explanation to my second colleague.
Thank you, madam. As friends, you have already studied some basic aspect of Lac Operon. Before going in further detail about Lac Operon, let me tell you two things. Lac Operon is controlled positively as well as negatively, both manner. So far you have discussed negative regulation of Lac Operon, and now onwards we shall go for positive regulation of Lac Operon. Positive regulation means what? The definition of positive regulation is only that positive regulation means some compound, in the presence of them, operon will start to operate, or operon will start to express. So that type of regulation is known as positive regulation. So positive regulation of Lac Operon is generally done by CAP protein, that is another regulator protein in Lac Operon. So let us see how it actually regulates the operon. In the presence of glucose and lactose, actually when culture is fed by both sugars, glucose and lactose, usually glucose is utilized preferentially first and lactose will be utilized later on, and this type of graph or this type of phenomena is known as glucose effect, or it is known as dioxy growth curve. Dioxy means this graph is having two lag phase, means actually bacterial cells are having only one lag phase and one log phase, while in this graph we can see that there are two lag phases are there. First lag phase is for glucose, and after glucose stopped, then second lag phase is for lactose, and then onwards it will switch over to lactose. So this is the graph which actually regulates Lac Operon. Yes, this is CAP or CMP regulation. This is the example of positive regulation. CAP means catabolite gene activator protein. It is also known as CRP or something like that. Usually what happens, CAMP, cyclic AMP usually binds with this CAP protein, and then CAP protein will binds to CAP site on Lac Operon. So cyclic AMP as shown in the structure, it is nothing but one derivative of adenosine monophosphate only, but it is converted to this form by enzyme adenylate cyclase. So this adenylate cyclase converts ATP to CAMP, and CAMP is needed for its further activity. So actually this CAP protein is actually catabolite repressor protein. It is also known as CAMP repressor protein. It is also known as CAMP regulator protein, and structurally it is homodimer, and each subunit is made up of 22,000 Dalton molecular weight. So usually this CAP site or CAP protein, it occupy the site which is known as CAP site in Lac Operon, but here without CAMP this CAP cannot directly bind to this site. So upon binding of this CAMP on CAP protein, it will induce certain conformational changes, as we can see in this diagram. That blue colored one is inactive CRP, that yellow colored one CAMP will bind to that, and then that inactive CRP will be activated by undergoing certain conformational changes. And now this CRP, activated or charged CRP, now it will bind to that CAP site which is just present beside the promoter. Okay, so if this CAP protein is bind to the this CAP site, and after that it will facilitate the binding of RNA polymerase, and in the absence of this CAP site, RNA polymerase will usually weakly bound to this site, and it may do some lower level of expression of Lac Operon. So for strong expression of Lac Operon, usually presence of CAMP is needed, and CAMP will charge the catabolite activator protein. Catabolite activator protein further facilitate the binding of RNA polymerase, and then Lac Operon will be continued. So friends, here we can see that in this diagram, not only the absence of repressor is needed, but also the presence of CAP protein is also needed equally to express all the genes of Lac Operon. This is the overall diagram of this Lac operator site. Here the green colored one is CAP binding site, where actually CAP protein is bind, followed by CAP protein, DNA covered by RNA polymerase. This is the actual site at where this RNA polymerase will bind. Okay, this is the promoter region for RNA polymerase, which is needed for transcription initiation. As we know that for transcription, -10 and -35, these two are the upstream site for are required or recognized for its initiation of transcription process. And એના પછી directly that will continue to transcribe latter part of Lac Operon. CMP લેવલ આપણે હમણાં થોડી વાર પહેલા જોયું પ્રમાણે કે ગ્લુકોઝની એબ્સન્સ અને લેક્ટોઝની પ્રેઝન્ટ that can stimulate the Lac Operon. So how actually it is possible? So that thing can be studied in by this diagram. Actually glucose અને એનું શું રિલેશન છે એ main that is given by this diagram, which is known as PEP-PTS system, phosphoenol pyruvate phosphotransferase system. Okay, so here PEP, as per this diagram, PEP is the main substrate. PEP is converted to pyruvate, phosphoenol pyruvate, and it will donate its phosphate group to E1, enzyme 1, and then there is one cascade will be formed by which phosphate will travel one by one step ahead. Okay, so first of all, PEP will donate its phosphate to E1, and E1 will be phosphorylated. E1 will donate its phosphate to HPR. So HPR, which is known as actually histidine permease or histidine protein. So that histidine protein, or sometimes we say it is also known as heat resistant protein. So that HPR will be phosphorylated and E1 will be dephosphorylated. So phosphate is simply conveyed to HPR. After phosphorylation of HPR, now HPR will donate this phosphate to E3 enzyme, which is present just beside the membrane, and again HPR will become dephosphorylated. Okay, so here sequentially phosphorylation, dephosphorylation is carried out, and there is one cascade will be formed. By that cascade, phosphate will travel from PEP to E2 side. E2 side, as it is shown in the diagram, it is one membrane spanning enzyme. It is present on membrane, and this enzyme, it actually regulates the entry of glucose. So when glucose enters, when glucose is available at E2 site, at that time finally this phosphate will be donated to glucose, and glucose will enter in the form of glucose 6-phosphate. But when glucose is not present, when culture is not having glucose in the medium, at that time what happen, this phosphate will not donated to glucose because glucose is not there. So in this case, this phosphate will be utilized. Here this phosphate will be make the activation of adenylate cyclase enzyme, which is also present just beside this E2 enzyme in the membrane. So adenylate cyclase will use that phosphate for conversion of ATP to cyclic AMP. So this is actually the process how in the absence of glucose this operon will turn on. So in the absence of glucose will make adenylate cyclase activation. Adenylate cyclase activation will cause CAMP level, and હમણાં જ આપણે જોયું એ પ્રમાણે CAMP level is actually required for CAP protein and then further activation of Lac Operon. So in the presence of glucose, neither adenylate cyclate will work nor CAMP will form, and if CAMP are not forming, then it direct it means that no CAP will be able to bind to CAP binding site. So actually Lac Operon is more or less controlled at this level. This is known as PEP-PTS level. There are so many such sugars are there, like glucose, mannose, maltose. These all are they all are known as PTS sugar. So actually it happens that in the presence of PTS sugar, non-PTS sugar like lactose, malibhiose, okay, this type of non-PTS sugar can never enter into the cell. They can never utilize by the cell, and that is actual base of this Lac Operon. So this is the cascade of phosphate group transfer from one place to another place, and at last phosphate will be donated to glucose 6-phosphate, and then it will enter into the cell. Now there are certain conditions are there in which Lac Operon will remain in off condition, that is it remains turns off. Okay, so which conditions are there to make this operon shut off or to make this operon switch off? Okay, so first condition is high glucose. હમણાં જ આપણે જોયું એ પ્રમાણે હાઈ ગ્લુકોઝ ઇનિબિટ્સ એડિનાઇલેટ સાયક્લેસ એન્ડ all the things. Okay, so high glucose will usually give negative impact on the Lac Operon. Second thing is low lactose. Low lactose is also having the same role. Low lactose means repressor will not detach from the site, so repressor will stop or it will inhibit the process. So in the presence of high glucose, in the presence of low lactose, now there are few conditions are there which are genetically altered condition. Means sometimes what happens, Lac Operon may not remain in its wild type state, but it may be mutated, like CAP mutant. Means that catabolite activator protein is not functionally working properly. So in that case, even if CMP are present, but they are unable to bind to this CAP protein, and it will not initiate the process. So CAP mutants are usually they are felt to express this Lac Operon. Next thing is promoter mutant. Promoter is the site at which usually RNA polymerase binds for initiation of transcription process of Lac Operon. So if this RNA polymerase binding site, that is promoter site, if it is altered, that is minus 10 base pair upstream or minus 35 base pair upstream, if this site has not been working properly, that means that will directly turn off the regulation. Means Pribnobox and Tatabox should be functionally active for active expression of Lac Operon. The next thing is the structural genes of Lac Operon, that is Lac Z, Lac Y and Lac A. If they will be mutated, then also Lac Operon will not be functionally active. Structurally it may express, but it may not be functionally fulfill all the functions which are expected from this Lac Operon. So in such conditions, sometimes cell may become Lac fermenter, sometimes cell may become Lac non-fermenter when Lac Z, Lac Y or Lac A, this type of cells, genes are mutated. And the last condition in this scenario when Lac Operon is remained off, that is IS. IS means that is super repressor mutant. Means that repressor molecule has been mutated in such a manner that even if lactose binds to that repressor, it will not respond to it and it will not detach from the site. So what will happen in such condition, whether lactose is there or not, it has no any impact or any difference in the expression of Lac Operon. Means this Lac Operon will remain permanently switched off. Means lactose cannot be able to turn on the expression. The same way, now there are few conditions are there in which Lac Operon remain always operated, Lac Operon always remain on. Okay, so these are the conditions like low glucose. Okay, low glucose will be there, then adenylate cyclase will be free. If adenylate cyclase will be free, then cyclic AMP will be formed, and all the remaining steps will proceed smoothly in this case. Second thing is IPTG. Presence of IPTG. IPTG is actually, it is considered as a gratuitous inducer of Lac Operon. Gratuitous inducer means such type of inducer which is not actually substrate. When substrate and inducer, both are two different things. At that time, inducer is known as gratuitous inducer. So for this operon, lactose is actual inducer as well as substrate also, while IPTG is only inducer, it is not the substrate. So IPTG is able to trigger on the expression of Lac Operon. The next condition is high lactose condition. Obviously high lactose will prove effective for expression of Lac Operon because all the genes will express. Repressor is unable to bind over the site. So all these sequences will leads to functional expression of Lac Operon. The next condition in which Lac Operon remain switch on, that is P, that is promoter wild type, Z, Y and A wild type. That means if all wild type genes are present, if there is no mutation is found at anywhere in any part of the Lac Operon, neither in structural part nor in functional part, then it will definitely leads to proper expression of Lac Operon. The next scenario, the next thing is I minus, that is repressor minus. Means if repressor is not found, if repressor is not at all found, then obviously Lac Operon will remain expressive, because lactose ni present hase ke nahi hoy, it will make not any impact on the operon, કારણ કે repressor જ નથી, એટલે there is no negative control is found at this that time. And a last condition je છે, that is known as O minus, or you may say that OC. OC એટલે એને કહેવાય constitutive mutants. Constitutive mutants means operator has been mutated in such a manner that now this operator will be it will be converted from inducible form to constitutive form. Means now it will constitutively express, and it will have not any impact of any inducer or repressor or positive regulator or negative regulator, and operator will remain continuously operative. Okay, so this type of mutation is known as OC mutation in Lac Operon. And the last type of mutation, that is O minus. O minus means operator itself, it is negatively mutated. So now operator ni absence ke present koi vastu feel thashe nahi, ane directly operator can be overcome by the RNA polymerase, and directly it will go for expression of further genes of Lac Operon. So these are the conditions in which Lac Operon will remain turned on, while previous conditions were there in which Lac Operon usually remain turned off. Okay, so altogether we can say that for overall functional expression of Lac Operon, group translocation should be there properly, that is adenylate cyclase and PEP-PTS system should be functionally active. Later on, Lac Operon itself should be remain active, cell should be able to utilize lactose, and all these things will lead to functional expression of Lac Operon. This is the summary part of this topic, that is what happen if glucose and lactose both are present, then no activator protein will bound in the presence of glucose because no CAMP will be there. So repressor protein will lift off the operator site, and usually in this condition Lac Operon will remain non-transcribed, that is it will not be transcribed to its functional product. Second scenario we have seen, that is when glucose is there but lactose is not there. So this is obviously having negative effect, means presence of glucose having negative effect on Lac Operon, and absence of lactose is also having the negative effect on Lac Operon. So altogether in such condition it will not undergo for any sort of expression. So again Lac Operon will remain turn off. The third condition is the same one, that is when glucose and lactose both are not there. So again slightly operon may be turned on because repressor protein repressor protein will block the site, but glucose absence will lead to CAMP cap activation. So altogether no transcription will be there, and at last when glucose is not found and lactose is found, this is the most ideal condition in which Lac Operon will remain expressive. So at last we can conclude that Lac Operon is an example of positive as well as negative regulation both. Negative regulation in that manner that absence of repressor is needed for functional expression of Lac Operon, while positive regulation in that manner the functional presence of cap protein is needed for functional expression of Lac Operon. So there are very few operons are there which are usually negatively and positively both way controlled. We can say that this strategy is actually regulation of control at transcription level, means here all the activities are regulated before transcription. Okay, so this is the one of the gene regulation strategy which occurs at transcriptional level. There are very other few operons are also there, like Tryptophan operon and Arabinose operon, or there are few other operons are there. Tryptophan has some different aspect, that is end product itself inhibits its own formation. So translational level control is there on transcription process and all. So these are the references for this topic. This topic is best given in Principles of Microbiology by RM Atlas 2nd edition, Microbiology by MJ Pazar and ECS Chain 5th edition, Microbiology Prescott, Harley and Klein 5th edition, Gene 8 Benjamin Levin, and Principles of Biochemistry Leninger 5th edition.
Thank you very much, Kunal bhai. As we have seen that regulation of a gene expression is very fundamental to any cell. આ એક એવી પ્રક્રિયા છે કે જે સેલને એનર્જી કન્ઝર્વેશન માટે ખૂબ મદદરૂપ કરે છે. જેવી રીતે આપણે આપણા ઘરની અંદર પણ એક રૂમમાંથી બીજા રૂમમાં જવું હોય તો અત્યારે જોરશોરથી પ્રચાર થઈ રહ્યો છે કે એનર્જી કન્ઝર્વેશન કરવી પડે તો આપણે સ્વિચ ઓફ કરી દઈએ લાઇટ અને બીજા રૂમમાં જઈએ તો બીજા રૂમની ચાલુ કરીએ, એ રૂમમાંથી બીજામાં જઈએ ફરીથી પાછા તો ફરીથી સ્વિચ ઓન કરીએ, એટલે જે જગ્યાએ જરૂર હોય એનર્જીની એ જગ્યા ઉપર આપણે યુઝ કરીએ અને એ રીતે આપણે એનર્જી બચાવી શકીએ. તો આવી જ રીતે સેલની અંદર પણ આ જ વસ્તુ લાગુ પડે છે કે ટ્રાન્સક્રિપ્શન અને ટ્રાન્સલેશન લેવલ જે છે, જેમ ફાઇનલી પ્રોટીન્સ બનાવે છે સેલના માટે જે જરૂરી પ્રોટીન્સ બનાવે છે, એ એન્ઝાઇમ હોય કે સ્ટ્રક્ચરલ પ્રોટીન્સ હોય, એ બધા પ્રોસેસ દરમિયાન ટ્રાન્સલેશન અને ટ્રાન્સક્રિપ્શનના પ્રોસેસ દરમિયાન ખૂબ જ પ્રમાણમાં વિપુલ પ્રમાણમાં એટીપી વપરાતી હોય છે. અને એ એટીપીનો જો સાવચેતીપૂર્વક યુઝ ના કરે સેલ તો સેલને પોષાય નહીં, એ બહુ કોસ્ટલી અફેર થઈ જાય. માટે આ બંને લેવલ જરૂરી છે. જેવી રીતે અત્યારે આપણે જોયું કે લેક ઓપેરોન છે એ પોઝિટિવ અને નેગેટિવ કંટ્રોલ બંને રીતે ગવર્ન થાય છે. સાથે સાથે આવા સેલની અંદર કેટલા ઓપેરોન હોય છે, કારણ કે ઓપેરોનનો મતલબ એમ થાય છે કે એક ક્લસ્ટર ઓફ જીન્સનો એવો સમૂહ કે જે એક પ્રકારનું ફંક્શન માટે રિસ્પોન્સિબલ છે. આ કેસની અંદર લેક ઓપેરોન જે શબ્દ વાપરીએ છીએ એનો મતલબ એમ થાય છે કે લેક્ટોઝના યુટિલાઇઝેશન માટે જેટલા જીન્સ છે એ બધા નોર્મલી પ્રોકેરિયોટ્સની અંદર એક સાથે એક સમૂહમાં આવેલા છે, બધા એક જ લાઇનની અંદર આવેલા છે. કોઈ જ વખતે એવા ઓપેરોન હોય છે કે જેની અંદર જીન્સ થોડા દૂર દૂર સિચ્યુએટેડ હોય છે, નહીં તો નોર્મલી પ્રોકેરિયોટની અંદર જીન્સ બધા એક સાથે આવે છે અને એનું રેગ્યુલેશન જે થતું હોય છે એ બહુ વ્યવસ્થિત થતું હોય છે. સાથે સાથે પ્રોકેરિયોટ્સની અંદર એક બીજો પણ ઇમ્પોર્ટન્ટ પોઈન્ટ એ છે કે એ જે ટ્રાન્સક્રિપ્શન અને જેવું ટ્રાન્સક્રિપ્શન પૂરું થાય છે એવું જ ટ્રાન્સલેશન શરૂ થઈ જાય છે. યુકેરિયોટ્સમાં આવું થતું નથી, કારણ કે ટ્રાન્સક્રિપ્શન પૂરું થયા પછી આરએનએનું પ્રોસેસિંગ થતું હોય છે, એનું મોડિફિકેશન થતું હોય છે, એનું સ્પ્લાઇસિંગ થાય છે. આ બધા પ્રક્રિયા પછી એ જે ટ્રાન્સક્રિપ્ટોમ છે એ સેલની બહાર નીકળે છે અને ન્યુક્લિયસની બહાર નીકળે છે અને ન્યુક્લિયસ પછી સાયટોપ્લાઝમની અંદર જ્યાં રાઇબોઝોમ્સ આવેલા છે ત્યાં આગળ રાઇબોઝોમ્સની અંદર ટ્રાન્સલેશન થતું હોય છે. એટલે પ્રોકેરિયોટ્સ અને યુકેરિયોટ્સનું ટ્રાન્સક્રિપ્શન અને ટ્રાન્સલેશન થોડાક રીતે જુદું પડે છે, પરંતુ બેઝિક પ્રિન્સિપલ્સ તો બધાનો સરખો જ હોય છે. હવે એક સેલની અંદર જેવી રીતે ઇકોલાઈ છે, ઇકોલાઈની અંદર લગભગ 4200 જીન્સ છે. હવે 4000 જીન્સનું રેગ્યુલેશન કરવું અને એ પણ એવું ઇકોનોમિકલી કરવું કે સેલને કોઈ જાતનું પ્રોબ્લેમ ના થાય, સેલનો ગ્રોથ વ્યવસ્થિત થાય, આ બધી એક ખૂબ અલ્ટિમેટ અને ફાઇન ટ્યુનિંગ માંગી લે છે, જે નેચરે ઇવોલ્યુશનના પ્રોસેસ દરમિયાન ઓવર અ પીરિયડ ઓફ સે મિલિયન્સ ઓફ યર્સ એવું સરસ રીતે ગોઠવેલું છે કે સેલનું કોઓર્ડિનેશન પૂરેપૂરું જળવાઈ રહે. એવું પણ જોવામાં આવ્યું છે કે સેલની અંદર અત્યારે સપોઝ લેક્ટોઝ નથી અને જેવું લેક્ટોઝ નાખવામાં આવે તો વિધિન ફ્યુ સેકન્ડ્સ, લેટ અસ સે વિધિન અ મિનિટ ઓર ટુ, પહેલો એન્ઝાઇમ મોલિક્યુલ બીટા-ગેલેક્ટોસિડેઝ સિન્થેસાઇઝ થઈ અને સેલને મદદ કરવા માંડી જાય છે, જેથી કરીને તરત જ સેલને વેઇટ ના કરવી પડે અને ટૂંક સમયની અંદર લેક્ટોઝનું યુટિલાઇઝેશન શરૂ થઈ જાય. એવી જ રીતે જેવો પણ એ સબસ્ટ્રેટ પૂરો થાય, લેક્ટોઝ ખલાસ થાય, તો તરત જ સેલને હુકમ થાય છે, ઓર્ડર થાય છે કે હવે જરૂર નથી એન્ઝાઇમ્સની, માટે એનું સ્વિચ ઓફ કરો. તો આ રીતે જે રેગ્યુલેશન જળવાતું હોય છે સેલની અંદર એ ખૂબ જ ફાઇન ટ્યુનિંગ સાથે સેટ થયેલું હોય છે. આ જ રીતનું યુટિલાઇઝેશન, આ જ રીતનું ઓપેરોન કંટ્રોલ બાયોસિન્થેટિક પાથવે માટે પણ હોય છે. જેવી રીતે હમણાં કુનાલભાઈએ જણાવ્યું કે ટ્રિપ્ટોફેન ઓપેરોન એક એક્ઝામ્પલ છે. ટ્રિપ્ટોફેન એક અમીનો એસિડ્સ છે અને એ અમીનો એસિડ્સનું જે બાયોસિન્થેસિસ થતું હોય સેલના દ્વારા તેમાં 4-5 એન્ઝાઇમ્સની જરૂર પડે છે અને એ એન્ઝાઇમ્સ એક સાથે સિન્થેસાઇઝ થવા જોઈએ આ ક્યારે સિચ્યુએશન જોઈએ છે કે જ્યારે સેલની પાસે ટ્રિપ્ટોફેન નથી. એનો મતલબ કે એન્વાયરમેન્ટની અંદર, મીડિયમની અંદર ટ્રિપ્ટોફેન અવેલેબલ નથી. At that time, cell has to biosynthesize these amino acids, and for which it will require 4 or 5 different enzymes. These enzymes have to be synthesized. In contrast to which, suppose if the cell has a tryptophan in its external environment, that is in the medium, then cell does not want these enzymes. These enzymes will be useless for cells. So it does not synthesize it, and it switches off the synthesis of these enzymes. આ જે ટ્રિપ્ટોફેનનું રેગ્યુલેશન થતું હોય છે એ રેગ્યુલેશન ટ્રાન્સક્રિપ્શનલ લેવલ તો ખરું જ, પણ એ ટ્રાન્સલેશન શરૂ થતાં પહેલાં એપ્લાય થતું હોય છે અને એને માટે એક લેડર પોલીપેપ્ટાઇડની આવશ્યકતા હોય છે. આ એક આખો બીજો ટોપિક થાય જેમાં આપણે ટ્રિપ્ટોફેનનું રેગ્યુલેશન ભણીશું પાછળના સ્ટેજ ઉપર. સો મિત્રો, આજે આપણે જોયું કે ઓપેરોનનું જે રેગ્યુલેશન જે આપણે સેલની અંદર થતું હોય છે, એક એક્ઝામ્પલ ઇકોલાઈનું લેક ઓપેરોન તરીકે ભણ્યા, કેટલું સરસ રીતે ગોઠવાયેલું છે એ બેશક દાદ માગી લે એવું છે. તો આપણે આજે આ બે મિત્રોને જે રાજકોટથી આવ્યા છે એમને આપણે આભાર માનીએ છીએ, અને વી આર ઓલ્સો થેન્કફુલ ટુ ધ ડિપાર્ટમેન્ટ ઓફ હાયર એજ્યુકેશન ફોર starting such a program on BISEC, that is SANDHAN, as well as the crew members, staff members of the studio who have taken lots of pains to telecast this program in an excellent manner. Thank you very much.